So good morning, everybody, and welcome to the webinar. The working title of the webinar is Activated Carbon and Subsurface Contaminant Degradation. I want to thank RPI and AST for the opportunity to present today. Of course, it helps if I can advance my slides here. There we go. So by way of introduction, my name is Peacock, and I'm the director of Microbac's Molecular Biology Laboratory in Oak Ridge, Tennessee. But the reason I'm here is because some of the work I've done earlier in my career I'm going to talk about. So just as some background, I spent 11 years at the University of Tennessee Center for Biotechnology, Environmental Biotechnology, and later at a place called the Center for Biomarker Analysis. And the research director of this lab was David C. White. And during that time, the center spun out a technology transfer laboratory that some of you might be familiar with called Microbial Insights. Most of our funding at CVA came from the Department of Energy and their natural and accelerated bioremediation program. And another major funder for us was the National Institute for Global Environmental Change. And the work at the university centered around subsurface microbial ecology applications, different types of applications in bioremediation, looking at global climate change, looking at applications for bioremediation. So after working at the research center, I took a job with Haley and Aldrich with some fantastic people as an environmental scientist. And while I was there, had the opportunity to work on just dozens and dozens of different types of contaminated sites. It was really a good opportunity. Beyond the research and then practical side of remediation, I had an opportunity to run the PACE Energy Fate and Forensics Lab. You folks, or some of you folks might remember that as microseeps. And for a few years now, I've been here in Oak Ridge, Tennessee at Microbac. And just I want to plug a Remediation Journal. So I'm on the editorial board for Remediation Journal. So anytime I get a chance to talk to a lot of practitioners out there, if you guys have any project data or research you'd like to publish in regards to this industry, please let me know because we're always looking for good articles. The other thing that I can't help doing is doing a shameless plug for Microbac. What we do is we can do qPCR for multiple bacterial targets that support bioremediation, etc. for reductive dechlorination, nitrogen cycle. We also do metagenomic characterization of subsurface microbial communities for bioremediation, water, wastewater, and we will be offering a surface swab test for COVID-19 here. We're pulling that one out in about a week. So if anybody has any facilities they need to verify that are clean, let us know. So for today, what about activated carbon? Does it hinder or help biodegradation of contaminants? Are contaminants adsorbed to activated carbon? Are they bioavailable? What's the story? So what we want to do today is we want to answer this in 3 parts. Essentially, part 1 is an introduction to subsurface microbial biodegradation of contaminants. Part 2, want to go over some of the research that was done. And I really can't believe this now as I was looking back at some of this 15 to 20 years ago using activated carbon in the subsurface. And then part 3, answer the question of where we are now and what could be next for activated carbon and microbial communities and understanding the mechanisms and whatnot goes on in those things. So microorganisms are the most abundant life on Earth, both in number of species and the quantity and weight of living organisms. They have a history spanning over about 3.5 billion years and have evolved to adapt to a wide range of environmental conditions and to survive on diverse sources of carbon and energy. So bioremediation is the technology that uses metabolic processes to degrade or transform contaminants so they are no longer in a harmful form. And what we do is we rely on these little guys to help us out quite a bit. They're kind of like the key to being able to have different things happen in the subsurface that, if they weren't there would be much, much more difficult. So microbial metabolism is how a cell makes a living. Actually, how it obtains the energy and nutrients that it needs to survive and reproduce. So for this example, we have a cell that uses organic material, right there, we'll put the organic material in, as a carbon and energy source, and also shows that we have other micronutrients that are required for life. As the carbon makes its way through the cell, it can be converted into cellular biomass or it can be processed through to carbon dioxide. And in this example, the cell is using oxygen as a terminal electron acceptor. However, on either end of this metabolism is the opportunity for specialized cells to act on contaminants. So we'll just drop those in there. As an example, a cell can use hydrocarbons as an electron donor or a carbon energy source, and then use chlorinated solvents as a terminal electron acceptor. So they can really help us out, but we've got to do some things for them. And so there's been a multitude of research on the characterization of various mechanisms and drivers of microbial biotransformation and degradation in the subsurface. Specifically, it's known as what makes the cell machine or the microbial community be able to operate. And that is, you have to have the availability of a suitable carbon energy source. You have to have the availability of a terminal electron acceptor. You have to have temperature and pH conditions conducive to cellular growth. And you have to have the availability of trace nutrients. You have to remove inhibiting metabolites or inhibiting compounds that may be in the subsurface that keeps them from growing or being able to do what they need to do. But you also need a suitable substratum for growth. So the old school way of thinking of microbial contaminant degradation was that planktonic cells kind of float or swim in the, air quotes, open groundwater. And if they happen to run into a contaminant, and if they have the capability, then they could degrade it. But, this way of thinking was way too limited, and it didn't accurately reflect what was going on in the subsurface. So the accepted paradigm now is that the subsurface microbes live as biofilms. Now, some of the advantage that biofilms have over planktonic living for microbes is that biofilms are cooperative and communicate. They have this thing called quorum sensing, and they can actually, not talk to each other in the sense that we do, but I mean, they can know what each other's up to. They can produce specialized metabolic functions. There's a physiological and social structure that depends on the types of microbes in the environment that are making it up, but they can react and live and make niches and do their thing. And the biochemical capabilities of a cell in a biofilm could be very different than that of that same cell that it's free floating, because now it has cooperation. It can do other things. There's this thing called lateral gene transfer also, which allows bacteria to share capabilities. The microbes are better protected in a biofilm. And so we can look at this as like a biological system. So the top picture on the right is of a biofilm, and you can notice that it's several cells thick. If I can say that right again, you notice that it's several cells thick. The bottom right is a picture of a biofilm recovered from a wastewater reactor that's colonized a small lava stone. So subsurface media have a hierarchical structure that controls physical processes of contaminant fate and transport. Phenomena at the very smallest scales can have a dramatic impact at the large scales. For example, processes at the cellular level occur at the micron scale, but processes can be observed at the field scale. Bacterial activity occurs at interfaces. So this is really important. This is something that I've learned, and you guys might know this already, but I think this is something that is really, really important when it comes to microbial ecology and bacteria and microbial communities in the subsurface, is that the activity occurs at interfaces. The interfaces include geochemical, say like at a aerobic, anaerobic interface, things are going to happen there, or it could be physical, like liquid to solid. So geological facies, things like that. Everything happens at interfaces. So when I was working as a researcher at the University of Tennessee, we thought that sampling groundwater for microbes might not be providing the full picture of what was going on in the subsurface. We thought that the gold standard for a sample would be to recover a solid subsurface material. However, collecting those types of samples was impractical. So think about it. It's a bit expensive to get a drill rig mowed to the site every quarter if you want to take a sample and see what's going on. So we thought that biofilms could provide a better data, and we didn't know what we should use as a sampling media. The microbial ecology paper referenced here presents the research we did investigating a glass wool media versus an activated carbon as a biofilm sampling substrate. The reason we used an activated carbon substrate was because I had a colleague named Carrie Sublette who ran a project where he used an activated carbon media in a bioreactor to treat lean streams of hydrocarbon contaminated wastewater. The results of that experiment showed the activated carbon substrate was more conducive to biofilm formation and supported a high biomass and a more diverse community composition than glass wool. So up there at the left-hand corner of the slide, you can see our little Rube Goldberg device that we use to house our media. And at the top is the picture of cells that have colonized the activated carbon. And then down at the bottom here, so here's the cells that you can see where we pulled this apart and put it under the microscope, confocal laser scanning microscope, took a picture of the biofilms that had attached themselves to the activated carbon. And then you can see here that even the glass wool had biofilm communities that started growing on them too. So you can see the strands of the glass wool here, hopefully, and the microbes that colonized them. So at about the same time as we were doing this work here, we were working at another site in Colorado where we deployed the activated carbon samplers. So we used this material here and put it in this site. This project was investigating bioremediation to immobilize uranium. And at this site, the major type of bacteria we identified on the activated carbon media was Geobacter acae, which is a well-known metal-reducing genus. One of the questions that came up was why Geobacter would colonize a substrate that did not include any oxidized metals. So it was thought at the time that Geobacter required oxidized metals as a terminal electron acceptor. So at the time, we thought that the reason Geobacter could colonize and grow on the activated carbon was because the activated carbon facilitated some type of electron transfer. But at the time, we'd had no idea what the mechanism was. It was just something that we thought, well, you know, these bugs shouldn't be growing here, but obviously they are, and why are they doing it? Well, there must be some way that, you know, Geobacter acae can, you know, dump a terminal electron acceptor. So if we then fast forward about 7 years, it turns out that Geobacter acae have something called electrically conductive pili, or nanowires, that they can use to transfer electrons to other cells or to activated carbon-like material. So Dr. Lovely's research group at the University of Massachusetts termed this capability direct interspecies electron transfer, or D.I.E.T. So here's the potential D.I.E.T mechanisms: electron transfer through electrically conductive pili, electron transfer through electrically conductive materials, that's our activated carbon, and electron transfer between electron transport proteins associated with other cell surfaces. So understanding D.I.E.T and the functionality it affords microbial communities associated with activated carbon is important. It opens up possibilities for added functions and capabilities for microbial community contaminant degradation. So Dr. Lovely terms these microbes as dieters, or he calls them E-communities, electronic or electric communities. So here's a little D.I.E.T example cartoon. On the left, we have a bacterial cell capable of D.I.E.T. In the middle is the activated carbon substrate, and on the other end is some PCE. In this example, the D.I.E.T capable cell dumps an electron to the activated carbon. The electron then travels along the activated carbon until it reaches the adsorbed PCE, where then it's reduced to TCE, and then, you know, can go on from there. So what do you think this means for contaminant bioavailability? If the cells don't even have to be anywhere near the contaminant in activated carbon to degrade it. So I want to say that again. What do you think this means for contaminant bioavailability if the cells don't have to be anywhere near the contaminant on activated carbon to degrade it? Now, this is a very simple example, but you might be able to imagine coupling of microbial biofilms with the capability of facilitated electron transfer. I would even take it further. And I would say in this scenario, for example, if we need a reductive dechlorinator, everybody talks about Dihalococoides, for example, Dihalococoides, Dihalogenamonas, Dihalobacter, Desulfidobacterium, all these bacteria that can, take Dihalococoides and Dihalogenamonas anyway, taking PCE all the way down to ethene. Oh, and you've got to have Dihalococoides at your site. If you don't have Dihalococoides at your site, you're stuck. But that may not be true. If you have activated carbon or like, the like material, the electron transfer can be facilitated through that conductor. So any cell that is capable of D.I.E.T could then take a PCE all the way down to ethene. But it's not only that, it's all kinds of other contaminants. These things that happen, we want to think that it's all so specialized, right? It is in a way, but Mother Nature has other ways that she has of working through things to be able to make things a little easier for everybody. So I don't know if you've ever seen any sites where you have complete reductive dechlorination and not any real measurable Dihalococoides, but I have. Not many, I haven't seen very many, but I have seen them. And I've always wondered, well, what the heck's going on? There's got to be something here. Anyway, I thought that was a really important observation. So let's talk a little more about activated carbons. Activated carbons are essentially porous, adsorptive, polymeric biocatalysts that support and facilitate the growth of microbial populations. Activated carbons provide a wealth of benefit to the microbial communities, including the absorption of organic contaminants and other organic nutrients. So what it does is it takes something that might be, especially if you're in an aquifer that's oligotrophic, these types of systems will grab all the organics out of it and hold it, right? But it doesn't hold it strongly enough that the bacteria can't get to it. They control contaminant metabolites and toxicity. So if the bacteria are taking care of the parent contaminant and then going to a metabolite, and the metabolite is toxic and normally it would kill off the microbe. Well, if it's on a system like this, the microbes, they can be automatically absorbed, keeps it away from the microbes so that the cells aren't being destroyed. It facilitates electron transfer, the D.I.E.T community, something we've just been talking about. There's a large surface area for microbial colonization. And there's a low shear for better attachment for these biofilms, and there's a protection from predation. So I'd like to talk about some specific contaminant degradation on activated carbon, but before that, I'd like to go over compound-specific stable isotope analysis, because that's kind of the results that we're going to be talking about here. I just want to give everybody a little bit of a refresher. So isotopes are molecules of an element with the same number of protons and electrons, but a different number of neutrons. So carbon-12 is about 99% of the carbon on Earth and has 6 protons and 6 neutrons, and that's termed light or stable. Carbon-13, on the other hand, is about 1% of the carbon on Earth and has 6 protons and 7 neutrons. The extra neutron does not have a charge, but it does have mass, and we can measure that mass. Now, carbon-14 is a radioactive form of carbon, and we don't use that outside the lab. So compounds with light isotopes, that means compounds like PCE that has light isotopes in it, will degrade faster than the PCE with the heavy isotopes. So the product remaining becomes isotopically heavier, and this process of isotopic change is called fractionation. It's really important to note here too, that absorption process, especially since we're talking about activated carbon, that absorption processes do not significantly change isotopic composition. There's been a lot of research on this. So here's an M&M's example of fractionation. So the blue M&Ms represent 12C or light carbon, and the yellow M&Ms represent 13C or heavy carbon. Going from left to right, the total mass of M&Ms becomes lower as they're being eaten or degraded, but the fraction of yellow M&Ms gets higher. This is essentially the same thing that happens to the contaminants as they are degraded. So if we look here, this slide shows the degradation via reductive dechlorination of TCE. And this is what we observe using on the left is what we observe using concentration metabolic product analysis. So the TCE concentration goes down. Oh, in fact, this is real. You can tell it's in the lab though, because I've never seen this exactly happen at a real site. But Maybe someone has, they've been lucky enough. But anyway, the TCE is going down. As the TCE goes down in concentration, the cis is being produced, it goes up, it hits a plateau, it starts to come down again. As it's coming down, the VC goes up, then comes down, and as the VC is going down, you have the ethene. So that's what we would normally see, if we were doing concentrations. On the right-hand side is the stable isotope data. So the TCE becomes more fractionated and then is gone. So it becomes fractionated, becomes heavier. So you see it going up there to the right, that little red one, red line. The cis DCE starts out lighter because remember, the first TCE that gets reduced is the light TCE. So that makes the cis DCE much lighter. And then that goes up, gets heavier and heavier. And the same thing starts over again for the vinyl chloride. And then the same thing happens for the ethene. And I have this silver bullet in here because the thing about using CSIA to track degradation in the subsurface, it's the closest thing I've found to a silver bullet because it's not going to be influenced by dilution or dispersion or anything like that. You're going to be able to tell if you have degradation. And you can't really do that with the concentration data, but you sure can do it with the isotope data. So this slide shows some example carbon-13 PC degradation data from a site where activated carbon was injected into the subsurface, and also there was an activated carbon in-well bioreactor operating. So the del of the PCE was minus 20 per mil, and the del of the TCE was minus 43.22 per mil. And when I first saw this, I'm like, wow, this is really kind of cool data. And I didn't know really where it had come from, or what the story was behind it. But we hardly, seeing fractionation, especially at a site where they had just started, just in a matter of a month or so, this kind of the field scale remediation efforts just really kind of boggled my mind. It was really, really cruising fast. So the bottom or the story here is that the isotope data showed at an activated carbon site that there was a huge amount and pretty fast amount of degradation of PCE to TCE. And the reason you can tell is the PCE is getting much heavier up there. Remember, these are negatives, so it's getting heavier as it goes up. And then the lightest material is being degraded first. So that evidence of the degradation of PCE as it turns into TCE becomes very, very light. So you see it's at a minus 43. And if we, we only did this once, but if we would have went down the timeline, we would show that, that whole thing would flip and flip again. But I don't even know at this site if we saw DCE or vinyl chloride, and that was all gone, or that was on the activated carbon and got taken care of right on the solids. So one thing that we can do with activated carbon is in-well biofilm samplers, is to preload them with a 13C labeled contaminant. So this is called stable isotope probing, or SIP. The way a SIP experiment works is to preload or adsorb a 13 carbon, a labeled carbon contaminant of interest to the activated carbon, and then deploy the sampler in a well and see if the contaminant degrades. The way that you know if it degrades is if the 13C carbon shows up in the microbial biomass and also the carbon dioxide or other things. So the way that we usually measure this is with the phospholipid fatty acids of cells, and this right here is kind of a illustration of a phospholipid. Here's your hydrophilic polar head. Here's your hydrophobic tails. This little sucker right here is the skin of the microbe, essentially. So what we can do is we can see if we have a labeled contaminant, say labeled toluene or labeled benzene, or something like that, to see if that'll be converted into this. If it will take that label into this right here. So let's see, this kind of shows it a little more. The slide shows how it works. On the left, you can see a cell with no 13C in the biomass right here, or in the lipids, or in the CO2. But when you add a 13C labeled contaminant, then the cell takes it up, it becomes part of the biomass and becomes part of the carbon dioxide. So back in 2004, we ran the first in-well 13C contaminant experiment in East Germany using toluene and benzene adsorbed activated carbon. So that work was published in ES&T. There's the reference for it there. The samples were deployed, I think, somewhere around 90 days. And once they were retrieved, we analyzed the phospholipid fatty acids. And this is a chromatogram of the phospholipid fatty acid profile of the bacterial community that was on the activated carbon sampler. Each of the peaks represents a different bacterial fatty acid. So the normal del ratios for PLFA are negative, but can vary. The chromatogram shows that some of the PLFA are highly enriched, meaning that in this case, the toluene was degraded and then biosynthesized into cellular material. And again, it proves that toluene that was adsorbed to the activated carbon was essentially degraded. So we did the same thing with benzene. So we had toluene and benzene. So this right here shows the results for the benzene. If you notice that there are fewer fatty acid peaks, and this usually means that there is a less diverse microbial community colonizing the activated carbon. Nevertheless, there is still very high del ratios that show the cells were degrading the benzene on the activated carbon. We've also had the opportunity to work with EPA and the Army Corps of Engineers on the McCormick and Baxter Superfund site in Stockton, California. The site was previously a wood treatment facility, and as you can see, there was a large creosote DNAPL issue. The question the EPA and Corps wanted answered was if the naphthalene was degrading under in situ conditions, which was methanogenesis. So we loaded the 13C naphthalene onto the activated carbon and essentially did the same thing as we did in East Germany. That is, we deployed the samplers in several wells and incubated them for a few months. Let them sit. So the Corps of Engineers had a strict quality assurance plan, and I remember that we were working with one of their environmental statisticians to develop an acceptance criteria for the results. There was a lot of boxes to check, but the project showed degradation of naphthalene. And there were signals in the fatty acids, there was signal in the carbon dioxide, and there was signal in the methane. So again, that showed that the naphthalene was degrading. The naphthalene that we had loaded onto the activated carbon was degrading. So when we did these types of tests, we would calculate rates of degradation for the contaminants that were on the activated carbon in the well samplers. But there was always a question of whether that was indicative of what was going on in the formation. So I'm sure that Scott Nolan at RPI has lots of rate data that he could share from some of their sites. I think all else being equal, the more activated carbon in the subsurface, the better chance for the development of these e-communities and dieters and biofilms that can degrade the contaminants. And if there's any modelers out there, what's the one compositional parameter where a small change can have a large impact in the subsurface? And that parameter is the organic matter. What the activated carbon is, in my opinion, is an engineered organic matter. It's that oikomene, right? It's that thing that gives life to the subsurface. So I've gone over some of the work I've done previously in my career, but obviously that's not all the contaminants that have been run with activated carbon. Here's a partial list of the contaminants that have been shown to degrade on activated carbon. So like benzene, toluene, naphthalene, we've got oxygenates, MTB, TBA, 1,4-dioxane, obviously the chlorinated solvents, pesticide, atrazine, RDX, and energetic, phenol. And also we should note that you know, in addition to the passive treatments, there's companies out there that have activated carbon in well reactors. There's going so those types of things. If this stuff it didn't work, people, I don't think they'd be doing it. And I wanted to include this slide one more time. It's not just activated carbon that makes for a successful remediation product. The cells need each of these things listed items in order for them to do their job. You have to have the availability of a suitable carbon and energy source. You have to have the availability of a terminal electron acceptor. You have to have the temperature and pH conditions that are conducive for bacterial growth, the trace nutrients, the removal of the inhibiting metabolites. Oh, and you got to have the microbial community. You can't, there's a lot of question about, or there has been a lot of question, at least back in the day, about whether bioaugmentation was useful or not. Well, I can tell you that I think that for the cost, it's useful, because it really doesn't cost a lot for, it's kind of rounding errors for these things. And it's something that can be, I think it's something that's needed, that can be used. So where are we now? I think the understanding the interactions between microbial communities and activated carbon and contaminant degradation can be described as a book. So we know what the first couple of chapters are, right? About the biofilms, about diet, and we know the ending. Yes, you can degrade contaminants on activated carbon. However, there are still chapters missing that would, seriously investigate the specific mechanisms of contaminant degradation. We don't know all the specifics about contaminant degradation on activated carbon. What are those things specifically? I think we have a good understanding about, first of all, about biofilms. We have a good understanding, or we're getting a better understanding through research like Dr. Lovely's research and whatnot on diet and dieters and e-communities. But even all that research is not particularly focused on how things work on these materials that allow electron transfer, and what that actually opens up for the possibilities of subsurface, remediation, for interactions, not just with contaminants, but even with, natural organic matter and things like that. So there's still a lot of room out there, I think, for even at the lab level, and I'm probably even more at the lab level than at the site level, but at the lab level of being able to understand exactly how all this works. Now, do we have to understand exactly how all this works in order to make it work at a site? I don't think so. No, but anyway, there's still that that's out there. So that is the end of my little presentation on activated carbon, and I guess I would be happy to answer any questions if I can. So I think I've got a couple of questions. Is the use of activated carbon something that would be supported by your research? Yeah, I think every site is different, but I think that yeah, I can't find where it would hurt anything. The only thing that I can see that all my research has done is shows that it would facilitate degradation of contaminants. Now, getting it in the right place, making sure that there's different types of activated carbon too. Not all activated carbons are created equal, but yeah. Here's a question: have you tested any compounds that are thought not to be degradable, such as PFOS or other recalcitrant compounds? No, I haven't done any of that. And in fact, a lot of the work, like I said, a lot of the work that I've done, this stuff's like 15 years old, 15 or 20 years old. So that was even before we even were worried about those chemicals. But I think that if we can find a mechanism, if there is a way, for instance, if there is a way for PFOS to be degraded, I would think that and I don't know if there's any bacteria that have the capability of doing it. But if there was, I would think that it could probably be facilitated, or better handled in a biofilm, better handled, with the absorption, or in a system setting, instead of just being a particular microbe setting, if that makes sense. Oh, the term, when I first mentioned the electrically conductive pili, what they first called them, at least this was back in the day, they called them nanowires. And maybe they've decided that they don't want to call them nanowires anymore, but I remember Dr. Lovely calling the electrically conductive pili as nanowires, and it was kind of a cool term. But they may not use that term anymore. It may just be conductive pili, electrically conductive pili. Can or does the AC fix micronutrients that are added to promote microbial growth? Certainly. Anything that's absorbable to the activated carbon is going to absorb, right? In fact, that was one of the things. If you have an oligotrophic aquifer, giving the bacteria a better chance to get established and whatnot, you would want that substrate for them to be able to attach to, where they could have a readily where they could readily access the nutrients. So do we have an idea of the number of different bacteria that would be typical to build a consortium of D.I.E.T? So that paper that we had from Dr. Lovely from 2017, where he did a review of D.I.E.T, he lists a whole bunch of the bacteria and the archaea that are involved in that. But what I would do, and I haven't had a chance to do this yet, would be to go to that paper. He has 132 references in that 2017 paper about D.I.E.T and the E communities. But then what we need to do is if you look at that paper and you go to the NCBI website or whatever, you can then look at all the papers that have referenced from 2017 to 2020 that have referenced that paper to see what other new critters have been found. And even Dr. Lovely says that this is still kind of all in its infancy. So even though it's been around for about 10 years now since they first discovered it, there's not all the research associates in the world that would be able to study every type of microbe to see if there's a capability for D.I.E.T. I think personally that it's probably the capability for D.I.E.T is probably a lot more widespread than what we would just assume. But that's just my personal opinion. It says, as far as tech vendors go, some recommend inoculation of AC prior to installation in situ. Can you confirm, based on your presentation, that this is the most efficient way to begin biodegradation and ensure necessary microbes are present for the process to be sustained, given all the other components are there? Like I said before, I think that I think that giving anything a head start is fantastic, and is probably very helpful. Earlier in my career, I would have said I didn't think so, but at this point, after we've seen the bioaugmentation, with reductive dechlorinators and whatnot be so successful, I can't help but think that, look, it doesn't cost much, it's a good box to check, and it probably does help. And it says, a follow-up clarification: relying on indigenous species to migrate and populate the AC can take a lot of time, provided the necessary species are present. That's true. I think that what we could do is we could definitely do some, I think that every site's going to be different in that respect, right? But again, if you can do it up front, go ahead. It's not gonna, the only thing it could probably do is help. It's not gonna help. Is it gonna be, in some sites, would it be substantially better? I don't know, we'd have to check. I don't have that data in front of me. And when we were doing this work, when I was doing this work, and we were trying to develop in-well samplers to answer specific questions, we weren't necessarily doing anything that was, for field scale bioremediation type stuff. It says, what is the evidence for gene exchange, particular to environmental remediation? I know that gene exchange occurs, but what are the specific instances? That's a good question. All I know, or what I can say right now, I can go in and send you some papers and everything, but all I can tell you is that the biofilms, that there is lateral gene transfer, and there's a lot of evidence for lateral gene transfer going on a lot more than we thought. We used to think that this stuff never happened hardly at all, right? But now we're finding it more and more. And what's interesting is that with the capabilities we have on the side of being able to do these metagenomic profiles and things like that, we're starting to build data sets that can help us answer these questions, even like down to perhaps down to the specific a specific commercial site, whereas we would never be able to have done that, 10 or 15 years ago. So is the biofilm and degradation process in the carbon essentially biologically regenerate the carbon's adsorption capacity? So we never really tested that directly, right? But my hypothesis is that's exactly what's going on, because these things are dynamic. It's not a static thing where something goes on, it absorbs and it's stuck. Like we said with the PCE, if you've got a microbial community and then you've got a PCE molecule or whatever that goes and absorbs, and then it gets knocked out. It's now ethene, it goes away. There's a space for more absorption. Now, what's the overall capacity for these systems? I don't know. I didn't study that, but it seems to me that is exactly what is occurring. That these things, it's not only this research that I've done, but it's some other research that we've done also that kind of shows the same thing in sediments and whatnot, where they had a biological mat that was placed, and they said that the life of this biological mat that had activated carbon would probably be on somewhere of several years, several tens of years or whatever. But then looking at it, and they would take cores of the mat and they would see that that it was never getting quote unquote full. It was never getting all the material staying on it. And the reason we did a microbial characterization of it, and there was all kinds of bacteria there that were eating hydrocarbons. So I guess I've seen data that show that these, at least in a lot of instances, and I'd say in my mind, it wouldn't bother me. I would think that they're regenerative. Is efficacy of introduced AC facilitated mechanisms affected by naturally occurring TOC? I think it would be, for sure. What those what that might be, I don't know directly, but I think it definitely would be. Well, I guess that's it. I'd like to thank everybody for hanging out, and if you have any other questions or anything, there's my email. I'd be happy to, if I have any papers or anything that you might be interested, send them your way, or try and find any other answers for you. So thanks a lot, and I'd like to thank again RPI and AST, and hope everyone has a good day and stay safe.